Since the body of vertebrates is mostly water, even a small wound could cause so much fluid to be lost over time that it would be difficult to maintain blood pressure to get oxygen to the brain, it would be difficult to regulate temperature, and it would eventually be life-threatening, which is why the blood contains thrombocytes, or in the case of mammals, platelets, which then can begin the coagulation cascade, starting a clot which blocks this hole. The earliest vertebrates possessed thrombocytes, which were cells. Here, note that they are elongate, and you can see these cellular thrombocytes in fish, in frogs, and in the turtle. In mammals, thrombocytes are not cells. The platelets, which are sometimes called thrombocytes, are formed in the bone marrow by giant cells called megakaryocytes under the influence of the hormone thrombopoietin. These cells break into 1,000 or 1,500 small pieces, each of which then is known as a platelet. So platelets are not actual cells, but fragments of this large bone marrow cell. These platelets are only found in mammals. How do the vesicles released by platelets or thrombocytes induce the formation of a clot, coagulation? It is complicated. It is a cascade of events involving multiple clotting factors where one clotting factor activates the next, which activates the next, etc. How could this complex cascade evolve? Interestingly, it is homologous to another cascade, an immune cascade, which would have been present in the earliest vertebrates. It is known by comparing genomes that there were not only gene duplications in vertebrate history, but two events which were likely duplications of the entire genome, or in other words, there were events where the descendants had double the number of chromosomes as the ancestral forms. This is known occasionally in species today, but it seems to have occurred twice between the jawless fish and the jawed vertebrates. In these events, the duplicate genes could then develop novel functions, and it seems that the serine proteases, which were once part of an immune cascade, then became incorporated into a new cascade which formed a fibrin clot at a wound site.